The topic that we shall be discussing is about antigen-antibody reactions. So these are nothing but the reactions that are usually used to identify either the antigen or the antibody. Initially it was like when we wanted to identify the causative agent related to a particular organism, that is a disease, what we used to do is we used to collect the sample. As we have seen in general principles of lab diagnosis, first what we do is we collect the samples, we process the samples, and then we carry out different procedures on the sample to identify the causative agent, such as going for the microscopic examination for the identification of the organism. Next is growth of the organism in the media, that is a selective media or enrichment media, depending upon which type of organism that has to be isolated. So these were multiple steps, and these are called as the classical experiments to identify the antigen or the antibodies. But now these multi-step procedures have been replaced with a unitary method to identify the antigen and antibody, and within a short period of time. Like when you want to identify the organism in classical experiment, it would take nearly about a day or weeks to identify the organism based upon its growth characteristic. But with the help of this antigen antibody interaction that are usually carried out in serological techniques, it is easy for us to identify either the antigen or the antibody related to the particular organism in a single step and within a short period of time. So the thing that we'll be discussing today is about the antigen antibody reactions, and it is mostly based upon the specificity of a particular antigen with a particular antibody. Now, as we know that during an immune response, whenever a foreign particle or the antigen enters the host responds it by producing the different types of antibodies which are usually specific to that particular antigen, and when they interact they usually form a complex which is called as antigen antibody complex, and this complex is only formed when there is a complementarity between the antigen and the antibody. Now further procedure or the further the binding of antigen antibody elicits the immune response to produce different kind of cells, ultimately eliminating the antigen from the human host. So this particular logic is mostly used in the laboratory to identify either the antigen or the antibody related to the causative agent of a particular infection. So this is the thing that we are going to discuss, that is the different antigen antibody reactions that are carried out in vitro to identify the causative agent related to a particular disease. So these are called as serological tests because it is mostly based upon the humoral immune response towards a particular antigen. It is called as the humoral immunity, and when this is used in the in vitro technique to identify the antigen, it is called as a serological reactions. So antigen antibody reactions are usually formed or usually takes place when there is a specificity in between the antigen and the antibody that can be seen in observable manner. And the purpose of this antigen antibody reaction serves a lot of things to us, that is because it usually takes place in vivo condition in the human body. When the antigen enters, it induces a production of humoral immune response, that is the antibodies are produced. So antibody mediated immunity is called as the humoral immunity. So the most important thing to eliminate the antigen in vivo is the elicitation of the humoral immune response. So the first purpose that is served by this antigen antibody interaction is the elimination of the antigen by eliciting the humoral immune response. Remember, it is not the antibody that eliminates the antigen, but it's a complex immune response towards a particular antigen that helps to eliminate the antigen, preventing the human host from the different types of diseases. Next, coming to the in vitro technique, how these antigen antibody interactions are very useful is because they help in diagnosing different types of diseases related to a particular organism. So each organism is able to elicit a particular disease in the human body. So depending upon that antigen, the particular antibody which is complementary to it is produced, and with the help of this complementarity, it is able for us to identify the specific antigen related to a particular that is the organism that is usually inducing an immune response in our human body. Next is in the epidemiological surveys. It helps in identifying the infectious agent or a new infectious agent that usually is causing the disease. So when a new organism is trying to develop the infection in the body, immediately the human host produces the antibodies against it. So identifying the antibodies helps us to identify the particular causative agent. So helps in placing the organism in a particular taxonomy, and also helps us to know the epidemiological surveys related to the infectious agents as well as the non-infectious diseases that are caused by the enzymes. Next is these techniques can either detect the antigen or the antibody. So relative to that particular disease only we'll be able to know which antigen is usually inducing the immune response or which antibody is going and interacting. So identifying either the antigen or antibody helps us to know the causative agent of that particular disease. Next is because this technique uses the antibody towards that particular antigen, these techniques are completely grouped together in the in vitro technique as the serological reaction. So the reactions that are usually done to identify the antigen or the antibody related to a particular organism in vitro technique is called as the serological reactions. So different from the classical experiments as we have seen in general principles of lab diagnosis. Now coming to the stages. Now what are the different stages that are required to see this antigen antibody interaction? So it is not a single step where you can see the interaction is taking place between antigen and antibody. Though they might take up immediately, that is they might react immediately, but they will not be seen in an observable manner until unless it is a secondary stage. So the stages of the interaction between the antigen and antibody can be classified into three major types. One is the primary stage, the secondary stage and the tertiary stage. So in the primary stage there is an interaction between the antigen and antibody, but you cannot see it in the visible form. There is no observable manner that helps us to identify the interaction between the antigen and antibody. But the interaction is very rapid. Whenever there is an antigen antibody that is present which are complementary to each other, they immediately react. So the reaction is said to be very rapid in nature, and it is reversible. Reversible means if there is no interaction, immediately the antigen antibody can get separated from each other, and because the antigen antibody interaction are held together by weak intramolecular forces such as van der Waals force, ionic force, etc., and not by the covalent interactions, these forces are said to be weak forces. But because there are multiple interactions that are taking place to hold the antigen antibody together, they usually forms a good complex between an antigen antibody. But there is no covalent interaction that can hold together the antigen and antibody, and hence you can see a reversible reaction, that is the antigen antibody getting separated from each other. And with the help of this primary stage, when you want to identify the reaction that has occurred between antigen and antibody, you can go with the estimation of free antigen or the bound antigen with the help of different techniques, that is the physical techniques or the chemical techniques, where you can use markers such as radioactive isotopes or the enzymes or the fluorescent dyes that are used to conjugate with either the antigen or the antibody. So with the help of this phenomena, you can identify or estimate the free antigen or antibody that are involved in the primary stage. But there will not be any observable manner, that is a positive reaction cannot be completely seen in the primary stage. The positive reaction is only possible in the secondary stage, where you can classify the secondary stage identification of antigen antibody interaction with the help of different techniques, which includes precipitation, agglutination, lysis in vivo mostly. When there is an interaction between an antigen and antibody, as I told you, there will be an immune response that will be developed towards the antigen and results completely in the lysis of the antigen. Neutralization in case of viruses or the toxins that are pathogenic in nature, again in vivo. And then you have complement fixation, where the complement, which are nothing but the serum proteins that brings about the lysis of the antigen in complex with the antibody. So all these lysis, neutralization, complement fixation test, immobilization, phagocytosis, all phenomena that are taking place in vivo condition, where we can see the observable manner or the positive reaction between the antigen and antibody in vitro condition is via the precipitation or the agglutination, and this can also take place in vivo condition. Now most of the secondary cases can move ahead only in vivo condition, resulting to the tertiary stage where complete elimination of the organism will be taking place. So tertiary stage only takes place in the in vivo condition, whereas secondary stage can be demonstrated in vitro condition. Primary can also be demonstrated where we can estimate the free antigen or the antibody when we are conjugating with certain markers such as radioactively labeled substances, enzymes, fluorescent dyes, etc. But tertiary stage only takes place in vivo condition, that is inside the host, where the organism can be eliminated via the neutralization process related to the virus infection or the infection that are caused by the toxins. Next is the destruction of the antigen, that is complete lysis of the antigen will be taking place using the complement mediated lysis. And then sometimes as we have discussed, this immune response can be exaggerated, leading to the tissue damage also. Apart from eliminating the antigen, they can also cause a tissue damage, which is called as hypersensitivity reaction. So all these tertiary stages are usually seen in vivo conditions where the antigen will be eliminated. Apart from eliminating the antigen, the other consequences or the other side of the immune response is leading to hypersensitivity or the immunological diseases such as autoimmune diseases. All together is clubbed together in the tertiary stage. So in order to identify the antigen or the antibody, the primary and the secondary stages are mostly used in vitro conditions, and they are called as serological reactions. Now coming to the general features, now how these antigen-antibody interactions or reactions usually takes place. So there are certain characteristics that have to be fulfilled so that these antigen-antibody usually forms complex with each other. First and the foremost is the specificity. The antigen and antibody has to be highly specific to form an antigen-antibody complex, and the specificity is only based upon the complementary shapes that are usually present on the antigen. So the antigenic determinants that usually elicits the antibody are nothing but the shape that helps to determine which type of antibody is going to be produced and goes and interacts with the antigen. So it is based completely upon the complementarity of the shapes between the antigen and antibody. If there is a complementarity, only there is a specificity and there is a reaction or the antigen-antibody interactions. Sometimes it so happens that cross reaction can also take place. Cross reaction is nothing but when an antigen enters, the antigen induces the production of antibodies. Now these antibodies, instead of going and reacting with that specific antigen, will go and react with the other antigen. Now this is only possible when there is a similarity between the two different types of antigens. So this is called as a cross reaction. Cross reaction, as we have discussed, can be both a positive or a beneficial role that can provide to the host, or a negative role. Beneficial role is in the vaccination of the smallpox virus, vaccination against the smallpox virus. Whereas the negative role that is given by the cross reaction is induction of the antibody towards the M streptococcal proteins can damage the heart muscle, because the heart muscle's antigen and the streptococcal M antigen is very similar to each other. The antibodies that are induced against the streptococcal M antigen, instead of going and reacting with that, they go and react with the heart muscle, causing the destruction of the heart. So that is a cross reaction. So sometimes it's very rare phenomena where you can see a cross reaction taking place. Otherwise, there is a highly specific reaction between antigen and antibody. And the next important thing is the entire molecule reacts. So the antigenic determinants which are present on the antigen induces the production of antibodies. They go and interact with each other, but when they are precipitating or agglutinating, it is complete entire molecule that usually precipitate or agglutinates. It's not only the antigenic determinant or the antibody, it is a whole molecule that usually reacts and participate in the response, that is antigen antibody response. Next is there is no denaturation of antigen and antibody. So as I told you, antibodies itself cannot cause the lysis of the organism or the antigen. It is only after they bind, they induce the immune response to produce different kind of cells to cause the lysis of the antigen. So in the first or the primary or the secondary stage, you will not see that there is a denaturation of either the antigen or the antibody until and unless there are different cells that are produced to eliminate the antigen. Next is the interaction is on the surface. So antigen usually carries these antigenic determinant sites mostly on the surface, and the antibodies are produced against the surface antigen. And in vivo condition, these antibodies are said to be protective in nature because they go and interact with the antigen, and they will not allow the antigen to cause the infection by eliciting the immune response. Next is the antigen-antibody interactions are firm because they are held together by a number of interactions such as van der Waals forces, ionic forces, hydrogen bonding, etc. They form a firm reaction between antigen and antibody, but they are reversible because they are not held together by the covalent interaction. And the firmness is usually known to us with the help of two concepts. One is called as the affinity and the avidity of the antibody molecule. The affinity of the antibody molecule is nothing but the interaction between a single antigenic determinant and a single antibody. So it is a single interaction between the antigen and antibody, which is called as affinity. So it is completely dependent on the closeness of fit between the antigen and antibody. And avidity is nothing but such multiple interactions between the different antigenic determinants on the antigen and different types of antibodies that are present or induced during the immune response. So it is a multiple interaction, whereas affinity is a single interaction between the single antigenic determinant and the antibody, whereas avidity is a multiple interaction between multiple determinant sites on the antigen and the different antibodies produced in relation to those antigenic determinants. Now here in the case of antigen antibody reactions, both antigen and antibody usually participate. It is not only antigen alone or antibody alone that usually interact. It is both the antigen and antibody. And in order to show the reaction between antigen and antibody, the antigen have to be multivalent antigen and the antibody should be a bivalent to multivalent antibody. So single determinant antigen and a single antibody cannot form the antigen antibody interaction. So there should be multiple interaction, and the antigen can have valency up to thousands or infinity, whereas the antibody should be a bivalent antibody or a multivalent antibody such as IgM antibodies. And the valency is very important to show the positive reaction between antigen and antibody in case of in vitro or serological reaction. So these are the general features that have to be satisfied by the antigen and antibody interaction to serve as a purpose to identify either the antigen or antibody in the serological reaction or in vivo where the antigen have to be completely eliminated. Now how do we measure these antigen antibody reactions in vitro conditions? So the measurement of antigen antibody interaction can be done either in terms of mass, units or liters. So mass is the complex that is formed. The amount of complex that is formed usually indicates the mass of that interaction, whereas the units or the titer is usually expressed in terms of antibody titer. Antibody titer is nothing but the highest dilution of the serum that shows a positive or observable reaction with in a particular test with that particular antigen. And the antibody titer completely depends upon the nature of the antigen, nature and the quantity of the antigen that is usually introduced in vitro technique, and the type of condition of the test, whether the physical condition or the chemical condition in which the interaction is usually taking place, that is the temperature, pH, etc. So the temperature and the pH have to be standardized before carrying out the technique. There are certain antigen and antibody interaction that can take place at low temperature only, and there are certain interaction that takes place only during the high temperature. So we have to know, we have to have a good knowledge of at what temperature and pH these interaction takes place. So as we know that antigen antibody interaction is highly specific. It depends upon the nature and the quantity of the antigen that we are using and the type and condition of the test that is usually performed in vitro condition. And there are two parameters that are usually considered when we are using this antigen antibody interaction in vitro technique. One is called as a sensitivity test and the other is called as a specificity test. Sensitivity test is used to identify either antigen or antibody in minute quantities, and in sensitivity test there is no false negative result, or there will be minimal false negative result that can be seen. So it is only used to identify either the antigen or the antibody, that is the quantification is usually done in sensitivity test. Detect even the minute picogram of the antigen or antibody can be done with the sensitivity test, whereas the specificity determines the specific interaction between the antigen and antibody. So here in the specificity test, you are going to identify the complex together, antigen and the antibody, whereas in sensitivity test you are identifying only the antigen or the antibody. So these are the two parameters that are usually considered when we are going with the antigen antibody interactions. Now, based upon the mechanism that is involved, based upon the antibody that is involved, you can classify the antibody into various types. So, in case of the antibody that is used in precipitin, it is a precipitation reaction. The antibody is called as a precipitin, and the relative antigen is called as precipitinogen. And if the antibody is involved in agglutination reaction, it is called as agglutinin, and the relative antigen is called as agglutinogen. So initially it was thought that a single antibody is able to carry out all these processes, that is precipitation, agglutination, etc. But by having a detailed study about these antibodies, we come to know that each reaction is usually carried out by different types of antibody depending upon the antigenic determinants that are used in that specific reaction. So they are corresponding antibodies towards that relative antigens that are usually used in carrying out that particular technique, whether it is a precipitation test or the agglutination test, complement fixation test, etc. So there are different types of antibodies that can mediate depending upon the antigenic determinants that are used in that specific reactions. Now there are different types of serological reactions that are carried out in the in vitro technique to identify either the antigen or the antibody. The first and the foremost is the precipitation reaction. Now the precipitation reaction is usually carried out by soluble antigen, or when a soluble antigen interacts with the soluble antigen interacting with a specific antibody in the presence of electrolyte, that is NaCl, at a standardized temperature and pH, forms a insoluble precipitate. Now sometimes it so happens that the precipitate that is formed usually forms a flocule, that it gets suspended between the two liquids. So this is called as a flocculation test. So if it is forming an insoluble precipitate that deposits at the bottom of the test tube, it is called as a precipitation reaction. And if it suspends as suspended molecules or precipitate suspended in a test tube in between, it is called as a flocculation test. So this precipitation test is given two names depending on how it gets precipitated and flocculated or gets sedimented. So it is a precipitation test or a flocculation test. And here the precipitation test can also takes place in the liquid media or in the gel media, where agarose is mostly used to see the precipitation reaction in the form of a band or a line, whereas in liquid you can see the insoluble precipitate that is getting settled at the bottom or as floccules in between the two layers of the liquid. Now when is this precipitation reaction taking place, and when you can see the precipitate that is formed? So it is only when there is an equal proportion of antigen and antibody that are mixed in a test tube, only then you can see the formation of a precipitate. So this was very well explained with the help of an experiment where a series of test tubes in which a fixed amount of antibodies were added. To this an increasing concentration of antigen was added, and the test tubes were seen for the that is the formation of a precipitate. The formation of precipitate was mostly seen in the middle test tube, where there was equivalent proportion of antigen and antibody. So this precipitate was absent in the previous test tube and the test tubes after the middle or the equivalent zone. So when a graph was drawn, it was seen that the precipitation is seen only in the middle test tube, preceded by an inclination of the graph which shows antibody excess, and next to the equivalent zone you can see the declination of the graph where you can see the antigen excess. So these two excess zones were called as prozone, where there is no formation of a precipitate or a flocculation reaction. So it is only at the middle test tube where there is an equivalent proportion of antigen and antibody that was able to show the precipitate. So this is called as the equivalent zone, and zone phenomena is mostly used to identify the equivalent proportion of antigen and antibody during during a precipitation reaction. And the formation of a precipitate was usually given to us by a scientist who is called as a Marak, and it is called as Marak hypothesis. So he said that in the relative proportion of or equivalent proportion of antigen and antibody, there is an interaction between antigen and antibody, and there is an alternative interaction between an antigen and antibody that causes a lattice, and this lattice usually precipitates, and which is absent in either the antigen excess or the antibody excess zone. So it is a lattice that is formed by the interaction between antigen and antibody, and there is an alternating antigen antibody interaction that forms a large lattice that usually precipitates or flocculates during this phenomena. So this particular phenomena holds good for the agglutination reaction too. So this is called as a Marak hypothesis, and it very well explains how the precipitation reaction takes place between antigen and antibody. Now coming to the applications, how do we use this precipitation reaction in serological techniques? So this precipitation reaction helps us to do the qualitative and the quantitative test to identify either the antigen or the antibody, and with the test we can even identify 1 microgram of the protein or the test antigen or the antibody in this precipitation reaction. That is very minute quantity of either the antigen or antibody can be estimated with the help of precipitation test. It is mostly to identify the antigen. Precipitation test holds it gives a very less sensitive test for the identification of the antibody. Next is this can be used to identify blood and the seminal stains, and next can also be used to know the food adulterants that are used in the food industries to identify the food adulterants that have been added, and it is less sensitive for the antibody, and hence we have to shift to other techniques to identify the antibodies, which are nothing but the agglutination test, complement fixation test, neutralization test, etc. So precipitation test holds good for identifying the antigens but not the antibodies. Now coming to the different types of precipitation or the flocculation test. The first thing is a ring test. Ring test, you can see a ring formed between the two layers of the solution, and here the procedure that is carried out is a layering of antigen solution over the antiserum in a narrow test tube, and the interaction takes place between the antigen and antibody, and a ring is formed between the two layers, and this technique is used mostly in the Ascoli's thermoprecipitin test or grouping of streptococci in Lansfield technique. Next is a slide test. As the name itself indicates, it is usually carried out on a slide where a drop of the antigen and the antiserum are mixed properly, and they are shaken and then mixed properly to form floccules. Example of this slide test is a VDRL test to identify the syphilic infection. Next is a tube test. This tube test is again used to identify the syphilis infection, and this is usually carried out in a series of test tubes. Apart from identifying the syphilis infection, you can also use to standardize the toxins or the toxides with the antitoxin. Next is the immunodiffusion or the precipitation in gels. So the previous techniques, this technique that we have studied are usually carried out in liquid. So it's difficult for us to identify the precipitate in liquid condition. So this has been converted to more sensitive test and the easier or rapid detection of this precipitation line in the presence of gels. So immunodiffusion is nothing but diffusion of the antigen or antibody in the gel to form a precipitate, and the positive reaction is usually seen as a visible as a distinct band of precipitate. It is more advantageous when compared to that of the liquid precipitation test, where the precipitate is usually seen in a liquid condition. Here we can see it is as a band on the agar that is mostly used, and the agar that is used is agarose, which is made of 1% of agarose in the particular test. And another advantage of this is it is stable, and we can stain it and preserve it for a longer period of time, and whenever we want we can identify this reaction. And next is it also indicates the identity between the different antigen or the antibodies. It also helps us to know whether there is a cross reaction that taking place, because there is sometimes cross reaction taking place, and it will confuse us in our diagnosis. So this precipitation in case of gels helps us to know about the cross reaction also that is taking place, and non-identity. If there is no identity, you can see as a crossing of line between the two precipitant bands. And here there are different modifications that are done in immunodiffusion. The first one is a single diffusion in one dimension, also called as the Ordine procedure, which is usually carried out in a test tube. And here they are usually in the gels, as we have seen, and the antibody is already incorporated in the gel and poured in a test tube, over which the antigen will be layered over, and the antigen diffuses towards the antibody in the gel and forms a precipitant band at a relative proportion of antigen antibody. And the number of bands that are formed also helps us to know the number of antigen that are present in a reaction mixture. So in case of liquid precipitation reaction, you can identify only one antigen antibody interaction, but in case of the immunodiffusion, that is which is carried out in gels, you can identify more than 10 antigens together in a particular test. So here the number of bands represents the number of antigens that are present. Next is a double diffusion in one dimension, also called as Oakley-Fulthorpe procedure, and here there is a plain agar between the agar that has been included with the antibody and the agar having the antigen. So both the antigen and antibody, if they are specific towards each other, moves towards each other and forms a line of precipitant in the plain agar which is present in the between. So the diffuses opposite to each other and they towards each other, and they form a precipitation band at the plain agar which is present in the between. Next is single diffusion in two dimension, also called as radial immunodiffusion. Radial means they form a circle or halo around the around the well. So here we have a slide on which we pour the serum, on which we pour the agar having the antibody and let it dry, and after that we make wells in the slide, and into the wells we add the antigen or the test serum that has to be tested. Now this antigen diffuses in the agar and forms a ring or band of precipitate depending upon the interaction between the antigen and antibody. The more the diameter that is formed indicates the more concentration of the antigen. So here the positive result is usually estimated as a ring or a band of precipitate around the well. Next is double diffusion in two dimension, also called as Octoloni double diffusion method. Here we have the plain agar on which the wells are usually made, and in the center well we add the antibodies, whereas in the wells that are usually surrounding the antibody well are incorporated with the antigen, and these antigen moves towards the antibody and they form a line of precipitate. Now this particular Octoloni double diffusion test also helps us to identify the cross reaction or the identity between the two antigen or non-identity between the two antigen. If there is an identity between two antigen, they fuse together to form a line of precipitate, and there is no identity between the two antigen, they cross away each other. Next is cross reaction can be seen as a spur formation. So this Octoloni double diffusion is used for a multiple test, that is on a single slide you can see multiple interaction taking place between a single antibody and different types of antigen. Next is the immunoelectrophoresis. So it is a combination of electrophoresis as well as the immunodiffusion. So in the electrophoresis we try to separate the components present in a compound. So take for example proteins, a compound completely is used over here, and this is electrophorosed to form different fragments, and towards this particular fragments we go for the immunodiffusion to identify a specific protein related in a particular compound. So electrophoresis is combined with immunodiffusion, and it is more sensitive when compared to the other techniques. So here the protein present in the serum will be electrophorosed into different components, towards which we go for the that is immunodiffusion. And here the testing normal and abnormal protein in serum and the urine sample is usually done. So in this technique what we do is first the protein that or the test serum that has to be identified or that has to go with the immunodiffusion is first electrophorosed at a right angle to the diffusion that is usually taking place. After that a trough is cut where the antibodies or the specific antibodies related to that particular protein are added. First after electrophoresis, when they get separated into compound, the immunodiffusion takes place, and you can see very well line of precipitate between a specific protein present in a compound with the antibody. So we will know towards which protein is the interaction with the antibody is taking place. So it is a multi-step again and helps to separate the compound and also knows help us to know towards which protein or the compound or the antibodies goes and interacts. Next is immunoelectrophoresis. Next is electroimmunodiffusion. So the immunoelectrophoresis can be speeded up by applying the electricity. So this immunofluorescence or immunoelectrophoresis might take about 18 to 24 hours to give us the reaction. But by applying electricity, this can be the speed of the reaction can be enhanced, and within 1 or 2 hours we'll get the result. So it is combining electrophoresis with the diffusion, applying the electricity. So in this electroimmunodiffusion, there are two methods that are mostly used. One is called as the counter immunoelectrophoresis, where the antigen antibodies are placed on a slide and they diffuse in opposite direction, and they usually form a band after diffusing in the opposite direction. Depending upon the specificity, they usually form a band. And the application of this counter immunoelectrophoresis is to detect the alpha fetoprotein in serum and antigens of cryptococcus and meningococcus in cerebrospinal fluid. And next modified form of this electroimmunofluorescence is the rocket electrophoresis, where the distinct band is usually seen in the form of a rocket, hence the name as rocket electrophoresis. Here the increasing concentration of antigens are placed in the well on a serum containing the antibody and allowed it to diffuse in the presence of electrophoresis. So here the band is usually in the form of a rocket, and hence it is called as a rocket electrophoresis. And the pattern is in the form of a rocket, and the modified form of this is a Laurel's two-dimensional electrophoresis, where the first technique that is carried out in this modified form is the electrophoresis and then with the immunodiffusion. Next important serological diagnostic technique is the agglutination reaction. So here in the precipitation reaction, we have seen a soluble antigen interacting with the antibody, but here in agglutination you can see the reaction is between the particulate antigen and the antibody in the presence of electrolyte against standardized temperature and the pH. They form a agglutinate or the visible clumps that can be seen. So these visible clumps, in order to be seen, they have to be a specificity, as we have seen in the precipitation reaction, that is the formation of a lattice that was the basic phenomena of the precipitation reaction takes place. The same holds good for the agglutination reaction, and this agglutination reaction is more sensitive when compared to that of the precipitation reaction. It helps in detecting even the antibodies, whereas precipitation reaction helps us to know about the antigens that are present. But here in the case of this agglutination, because particulate antigens are mostly used, it also helps us to detect the antibody. The same zone phenomena holds good for the agglutination reaction, where which states that equivalent proportion of antigen and antibody reacts to give rise to a visible clump or agglutinate to give the observable manner positive result. And here the incomplete or monovalent antibodies do not form agglutination reaction. So the antigen should be a multivalent antigen and the antibody should be a bivalent antigen or a bivalent antibody or a multivalent antibody to give rise to a agglutination reaction. Now it has a lot of application in the serological techniques, that is the agglutination reaction. This can be carried out in the slide agglutination, where a slide is used and the antigen and antibody the anti-serum is usually added drop-wise in equivalent proportion and mixed properly with the help of an applicator stick and can be seen as visible clumps. In order to be sure that the clumps are a positive result, you can also view it under the microscope. So remember, equivalent proportion of antigen antibody has to be added on a slide or on a kit or on a tile and then mixed properly and seen for the result. The agglutination occurring after a minute or 2 minutes cannot be taken as a positive test. It is only within a minute you can see the interaction. That gives a positive interaction. And this slide agglutination test can be used for a variety of tests to identify the blood groups, to identify the organism causing typhoid, etc. Apart from that, you can also go ahead with the tube agglutination test, which is a serological diagnosis for typhoid, brucellosis or typhus fever. So here in the tube agglutination test, the antigen anti-serums are mixed in equivalent proportion and helps in identification of the either the antigen or the antibody in the test tube by the formation of agglutination or the clumps. Mostly in the typhoid, it is used to detect the H antigen or the O antigen. H antigen is a flagellar antigen that usually forms cotton wool type of agglutination, and then you have O antigen that gives a different result in the case of the tube agglutination reaction. So based upon the pattern of agglutination that we get, we can identify which antigen is mostly involved in the infectious or the disease process. Next is the antiglobulin or the Coombs test. So here this Coombs test is mostly used to identify the Rh antibodies that are usually produced in disease which is called as erythroblastosis fetalis. So Rh antibodies, for the detection of Rh antibodies, the antiglobulin or the Coombs test is usually done. The examples of the agglutination reaction using the red cells, that is the erythrocytes or the RBCs, are Paul Bennett test and the cold agglutination test. In the Paul Bennett test, it is used to identify the patient suffering from infectious mononucleosis, where the sheep RBCs are mostly used that are sensitized with the antibody, and here the sheep that is the antibodies usually interacts with the ox red that has ox RBC that has been sensitized with the RBC. Examples of the agglutination using the RBCs or the erythrocytes, there are two techniques that are mostly used where RBCs are used to detect the agglutination reaction. It is a Paul Bennett test, and next is a cold agglutination test. Paul Bennett test is used to identify the antibodies towards the infectious mononucleosis, where the sheep antibodies are usually adsorbed onto the ox RBCs. So positive reaction can be seen in the form of a clumping, whereas in the cold agglutination test, the name itself indicates the cold agglutination takes place at a very low temperature at around 4 degrees centigrade. So here the positive is seen in the mycoplasmal pneumonia, where the agglutination reaction takes place at 4 degrees centigrade and it can be reversed at 37 degrees centigrade. Next is the antiglobulin or the Coombs test. So here the antiglobulin or the Coombs test is mostly used to identify the anti-Rh antibodies. So towards the Rh factors, antibodies are usually produced. In case of erythroblastosis fetalis, the disease or the disorder associated with the Rh negative female. So here we are trying to identify the Rh negative antibodies with the help of a Coombs test. So it is called as an antiglobulin test because these antibodies will not be able to fix or will not be able to agglutinate the RBCs. So the serum contains incomplete antibodies that sensitizes the Rh antibody. So the RBCs are sensitized with the incomplete antibodies. Now these RBCs that have been sensitized with the incomplete antibodies are washed to remove any unnecessary protein, and they are treated with the Coombs reagent containing the antibodies towards these incomplete antibodies that are sensitized the RBCs. Now they are able to agglutinate and gives a positive reaction. So this is usually used to identify the anti-Rh antibodies that are seen in the erythroblastosis fetalis. And this Coombs test can be carried out as a direct Coombs test or the indirect Coombs test. So in the direct Coombs test, it can be seen in vivo condition where the Rh antibodies are taken from the fetus, that is the baby that is born, and trying to identify these Rh antibodies that are causing the destruction of the baby. So here the patient serum is taken and is sensitized with incomplete antibodies, and they are carried out with the further procedures by adding the Coombs reagent to identify the agglutination reaction. In case of in vitro technique, we can sensitize the RBCs directly with the incomplete antibodies, and it is usually used to identify different types of Rh antibodies that are involved in different types of disease or disorders. Next is a passive agglutination test. So here the passive agglutination test indicates that in case of both precipitation reaction and the agglutination reaction, antigen antibody is very essential. But in case of precipitation reaction, we have seen soluble antigen taking part in the precipitation reaction, whereas in the agglutination reaction, it is the insoluble or the particulate antigen that is taking part in agglutination reaction. So the disadvantage of the precipitation reaction, which cannot detect the antibodies, can be converted to advantageous procedure by mixing the soluble antigen with a carrier molecule, which is called as which are nothing but the RBCs or the latex particles or the bentonite, to completely change them to the particulate antigen. Now these particulate antigen can be used in detection of the antibodies that are involved in the agglutination reaction. So for converting the soluble antigen to the particulate antigen, certain carrier molecules are mostly used. They are nothing but the RBCs. It can be from the human or the sheep RBCs, or you can make use of the latex particles, which are nothing but polymeric molecules that are artificially synthesized, or you can also make use of certain chemicals which are called as a bentonite. So here the soluble antigen with carrier molecule converts itself to the particular antigen, can be used for the detection of antibodies. So the protein antigens that are used can be directly, polysaccharide antigens can be directly used, whereas the protein antigen, for identifying the protein antigen, they have to be mixed with the tanned red cells. And the best example of identifying the antigen or antibody is a Rose-Waller test of the passive agglutination reaction. So here the Rose-Waller test is mostly used to identify the RA factor, which is usually involved in rheumatoid arthritis. RA factor is nothing but the antibodies that is produced against the human immunoglobulins, that is the IgG or the IgM molecule that is involved in autoimmune diseases. So rheumatoid arthritis is an autoimmune complication that is seen in the host. So in this particular patients, the patient produces antibodies against the self antibodies. So these RA factor are not able to agglutinate the RBCs. So in order to make them agglutinate, the RBCs have to be sensitized with the globulin or the human immunoglobulin. Now once they are sensitized with the human immunoglobulin, the RA factor is able to agglutinate them. So depending upon this agglutination reaction, you can identify the RH factor, that is RA factor, that is involved in the passive agglutination reaction, to identify the RA factor involved in rheumatoid arthritis. So when making use of the artificial substances such as latex agglutination test or using the latex particle, you can carry on different techniques, that is to identify human chorionic gonadotropin hormone or the RA rheumatoid arthritis or CRP reactive proteins, etc. So this is about the agglutination reaction, that is a visible reaction in the form of clumps, the reaction between antigen and antibody using the RBCs. Next is a complement fixation test. So complement fixation test means the use of complement proteins. Now as we all know, complement proteins are nothing but the serum proteins that usually circulate along with the antibodies. They are very heat labile, and they carry out a reaction which is called as a cascade reaction in order to form the pores or the lysis of the antigen along with the antibody. So different pathways by which this complement protein which are inactive in the serum can be activated, and it usually completes the action of the antibody in eliminating the antigen. So that is called as a complement mediated lysis. So these complements, after a cascade of reaction, they form a membrane attack complex that attacks the membrane of the bacteria and causes the pores and kills the bacteria along with the antibody. So this logic is mostly used in the complement fixation test. That is, when the antigen antibody interaction takes place, it is said that a complement has been used up or it has been fixed. When the complement has been fixed, that means there is a lysis of the antigen. So in this particular technique or in vitro technique, what we do is we identify whether there is any involvement of the complement or not. The presence or absence of complement help us to know the positive or the negative result, whether the antibody specific towards that particular antigen is there or not. So this complement fixation test helps us to detect 0.04 mg of the antibody nitrogen and 0.1 mg of the antigen that is causing the infection in our body. So here the negative CFT is usually indicated in the form of a lysis, and positive complement fixation test is indicated by no lysis. So there are two set of tests that are usually done in this particular complement fixation test to see whether the complement protein has been fixed or not. So here in the first test, that is the interaction between we require 5 components over here. One is an antigen, antibody, we require the complement protein, and next we require the sheep RBCs and the antibody against the sheep RBCs. So antigen can be any antigen towards which the antibodies are usually going and acting towards. The antibody that we are going to select has to be treated so that no complement protein is attached to it. And the complement protein that we are using in this particular technique will be from the guinea pigs. And again, to this we have to see that there are no antibodies associated with this complement protein. And the sheep RBCs are acting as an indicator system along with the antibodies against the sheep RBC, which are called as the AMBO receptors. So in this particular test, first what we will be doing is antigen and antibody are incubated for a certain period of time so that the interaction takes place. Then we add the complement so that if there is a specific antibody towards that particular antigen, complement will be fixed up. Now this step helps us to know whether the complement has been fixed or it is free to react with the sheep RBCs and the antibody. So here complement next we add is the sheep RBCs along with the antibodies. So the result can be estimated depending upon the lysis. If there is a lysis, that means the complement was not fixed up, which means the antibody is not specific towards the test antigen. So if the complement is free, it is able to cause the lysis of the sheep RBCs. Lysis indicates a negative complement fixation test. In a positive complement fixation test, if the antibody is specific towards the antigen antibodies complex is formed and the complement will be fixed, and it is not free to lyse the sheep RBCs along with the antibody. So no lysis means a positive complement fixation test. So here the example of the tests that are mostly used to carry out the complement fixation test is a Wassermann test for the detection of syphilis. Now there is certain modification of this complement fixation. There are some antibody that do not fix the complement of the guinea pig. So alternative form of complements that are mostly used. So in case of avians, mammalians such as horses and cats, they do not fix the guinea pig complement. So in that case we make use of an alternative complement, and the test procedure will be same, but after the initial reaction that has taken place, we add the standardized serum which contains the complement. So the positive result can be estimated as the hemolysis indicate a positive test, and in case of the conglutinating complement adsorption test, the horse anti-serum containing the complement is mostly used. Here also the test is mostly used to detect the type of reaction where the guinea pig's complement cannot be fixed. So here alternative form of complement, that is a horse complement which is non-hemolytic, is mostly used. So indicator here is a indicator antigen here used is a sensitized sheep erythrocyte along with the bovine serum. The bovine serum has a component which is called as conglutinin that is able to agglutinate the sheep RBCs. Now, in the case of this conglutinating complement absorption test, the sensitized cells, that is agglutination of the sensitized cells will not take place. So, indicates a positive reaction. Now coming to the other complement dependent serological test, here you can identify the bacteria that are involved in the in vivo pathogenesis, that is the Vibrio cholera or the Trypanosoma pallidum, and the test is said to be immune adherence. Here there is a mixture of the antigen antibody along with the complements are usually added, and if there is an immune adherence, there is a formation of a complex between these three. It is called as the immune adherence, and helps us to identify whether there is any specificity between the antigen and antibody. In immobilization test, we act the active trypanoma organism involved in syphilis, antigen antibody complement, and the antibody will immobilize the trypanoma. So based upon the immobilization of the trypanoma organism, we can identify the positive or the negative reaction. Next is a cytolytic or a cytocidal test that is used to cause the lysis of the Vibrio cholera. That is a cholera vibrio that causes cholera. Here the antigen and antibody are mixed together, and the antibody along with the complement causes the lysis of the Vibrio, which is nothing but cytolytic or cytocidal test. Next test of serological reaction are the neutralization test. So neutralization is a phenomena that is usually carried out to eliminate the virus or the toxins from the body. So agglutination and precipitation can be done to identify the bacteria and other organism, whereas virus usually causes the pathogenesis in the human by making its DNA enter into the human host. So it is to prevent the viral infection, what we have to do is we have to neutralize the virus. Neutralizing the virus is nothing but making the receptor unavailable for the virus to go and interact, blocking the receptor where the virus usually goes and binds. So similarly, toxin also have the pathogenic effect only after it enters the human cell. So we are preventing the toxin and the virus from entering the cell by not allowing them to bind to the receptor on which they usually bind. So they are called as neutralization of viruses or the toxin. So there are two different tests that are involved in neutralization test. It is called as a viral neutralization test, and the virus pathogenesis can be known by growing a lawn of bacterial cell, and then inoculating the bacteriophages on the bacterial cells. So if there is any specificity between the organism and the virus, they usually causes the lysis of the bacteria and forms a plague or a zone of hydrolysis around the colonies. So this indicates a plague that is a positive test for the virus pathogenesis. So we can add an anti-serum to that particular test and inhibit the plagues that are formed on the test. So this is called as a plague inhibition test, that is not allowing the virus to cause the pathogenesis to the bacteria and cause the lysis. So with the help of plague inhibition test, we can identify the viral neutralization, that is adding a specific anti-serum to the viruses. That is the anti-serums are nothing but the antitoxins or the neutralizing antibodies towards that particular viruses. So prevention of the plague formation indicates a positive reaction. And similarly, toxin also causes the pathogenic effect inside the human, and there are exotoxin and endotoxins that usually are pathogenic, and here these toxin neutralization test can be done either in vivo or in vitro. In case of diphtheria toxin, in vivo condition, we can detect by injecting a small amount of diphtheria toxin intradermally, and the antitoxins that are usually present can be detected via the skin test. And similarly, the Schick test is also done to identify the neutralizing antibody or the antitoxin present in the human. So a small amount of antigen will be introduced, and to detect whether antitoxins are there, we'll be knowing whether there was a previous immunization or the previous contact with that particular disease. And in vitro condition, we will go with the anti-streptolysin O test, where the hemolytic inhibition usually takes place. So anti-serum is added to the streptolysin or the strepto organism, where these organisms are said to lyse the RBC. So by adding the antiserum, we are preventing the hemolysis that is taking place. So this is a in vitro test to identify the anti-streptolysin O test. Next is the opsonization. Opsonization is nothing but enhanced phagocytosis, and this is related to the organism that are usually engulfed by the polymorphonuclear leukocytes, where these macrophages or the neutrophil engulfs the organism, and they process and present to the T cells, and helps in the elicitation of the humoral or the cell mediated immune response. And this opsonization can be enhanced when the complement and the antibodies are together used to present the antigen to the macrophages, which can be easily phagocytized by the macrophages. So this opsonization is used to know the resistance of the disease or disease during a disease process. So here it can be expressed in the form of an opsonic index, which indicates the phagocytic activity of the patient's blood to the bacterium to the phagocytic activity of the blood from the normal individual. And this can be estimated by the average number of phagocytic bacteria that has engulfed the organism by to the ratio of the average number of the phagocytic bacteria to the polymorphonuclear leukocyte that have engulfed the bacteria. So this helps to know the resistance of the human body towards the antigen during the course of an infection. Next is immunofluorescence. Immunofluorescence, that is the previous tests that we have studied are though sensitive, but they are not to that extent that they can detect even the minute quantity of the antigen or the antibody, and it is easy for us to identify the antigen within minutes or seconds with the help of immunofluorescence technique. So fluorescence is a phenomena where particular dyes called as fluorescent dyes are able to take up one one wavelength of light and emit another wavelength of light, which are used to identify the interaction between the antigen and antibody. So there are different dyes that can be used in this immunofluorescent technique that are conjugated to either the antigen or the antibody. When there is an interaction, these fluorescent dyes usually fluoresce and gives away a different light against a dark background. So it is a orange red color or a green color that can be easily identified under the fluorescent microscopy. So the fluorescent phenomena is the technique that helps us to identify the interaction not only in vitro condition but also inside the human body, where there is a development of a particular disease or a cancer inside the human body. So these conjugated antibodies goes and interacts with that particular antigen, and they fluoresce. When they fluoresce, it is easy for us to identify in which part of the body is the infection present and in which part of the body is the cancer developing. So that is combining the fluorescent technique with the histological technique. So combining the serological technique with the histological technique to identify. So this has a very well advantage in the serological reaction, which helps to identify either the antigen or the antibody. And here either the antigen or the antibody can be conjugated with the fluorescent dye. So in the case of direct fluorescence, you have only two components. One is the antigen or the antibody that can be conjugated and mixed in a mixture, reaction mixture. If there is any specificity, you can see the fluorescence phenomena giving away a different amount of light. They take up a white light and they give up a different amount of light. And here in the indirect fluorescent technique, mostly used to identify the Trypanoma pallidum, which is a very sensitive organism that cannot be easily identified by the other techniques. Here in this particular technique, what we do is we have first combination of antigen and antibody. That is a reaction taking place between the antigen and a specific antibody, and towards this specific antibody we have another antibody to which the fluorescent will be conjugated. So it is easy for us to take a simple conjugated antibody to detect different types of reaction that takes place between the antigen and antibody. So you need not make use of different types of antibody conjugated against different antigen. If you have this conjugated antibody towards a particular antibody that is specific to it, is easy for us to make use of a small number of conjugated antibodies to detect more amount of antigen antibody interaction. So it is called as an indirect fluorescence because the antibody that is directly interacting with the antigen is not conjugated with the fluorescent dye, but it is rather the antibody against the antibody that has interacted with the antigen. So there is a second antibody that has been conjugated, and this second antibody has receptor on the Fc portion of the primary antibody. So that is called as the indirect fluorescence used to detect the trypanosoma pallidum. Another method can also be used in immunofluorescence, which is called as the sandwich fluorescent technique, where the antigen will be sandwiched between two types of antibodies. So first there is an interaction between antigen and antibody. Towards this antigen antibody complex, you have another antibody going and interacting which is already conjugated with the fluorescent dye. So it is called as sandwiched because the antigen is sandwiched between two different types of antibody. One specific antibody towards that antigen and another antibody that is conjugated with the fluorescent dye towards this immune complex. So that is called as immunofluorescence technique. And it is more rapid, more sensitive, helps in detection very well, easily detecting either the antigen or the antibody. Now, apart from this, there is a modification by using different types of markers that can be conjugated to either the antigen or the antibody. They are called as radioimmunoassay. Here, the marker that is used is a radioactive isotope that act as a label. So, in the fluorescent or immunofluorescent technique, fluorescent was a label that was used to conjugate with either antigen or antibody. Here in case of radioactive isotope, the radio labeled substances are usually conjugated to either antigen and antibody. So it is based on a binder ligand assay where the ligand is an antigen and the binder is an antibody which goes and interacts with the antigen, and to this binder we usually conjugate with the radioactive isotopes that when goes and interacts with antigen, and when there is a positive interaction between antigen and antibody, it is radiographed, and depending upon the intensity of the radio activity that is produced, we can identify whether there is an interaction between antigen and antibody or not. So this radioactive radioimmunoassay helps us to measure about picograms of the antigen or detect a very small amount of the antigen. Also helps in detecting the haptens too. So mostly the application of radioimmunoassay is to detect the hormones or the drugs, tumor markers inside the human body, and IgE, which is mostly related to hypersensitivity reaction, and viral antigen. So the antibody towards these particular antigen, that is a ligand, can be conjugated with the radioactive isotope and made to interact with this, and the radioactivity is usually measured. And this radioimmunoassay is usually based upon a competitive interaction between a labeled antigen or the labeled antigen and unlabeled antigen. So here the antibody amount will be fixed, whereas the labeled antigen will be added along with the unlabeled antigen. So there is a competition between a labeled antigen and the unlabeled antigen towards a particular antibody. The more amount of specificity between these two, that is whether it is a labeled antigen or an unlabeled antigen, can be known with the help of the radiograph, the radioactivity that is usually produced. More amount of radioactivity indicates less specificity between the antigen and antibody, that is a test serum, unlabeled antigen. Less amount of radioactivity indicates more amount of interaction between the unlabeled antigen and the specific antibody. So here the antigen is usually labeled with the radioactive isotopes. The more specific method, apart from the radioactive radioimmunoassay, where there is association of radiation hazard, we can now shift or move to the conjugation of antigen and antibody with the help of enzymes. So these are called as enzyme immunoassay. Now, as we all know, enzymes are the one that usually mediates a chemical reaction, where the enzyme usually reacts with the substrate to give rise to a product. So here the enzyme enzyme that is used interacts with the substrate that can be added at the later part of the test, and depending upon the product that is formed, we will know whether the interaction between antigen and antibody has taken place or not. So this enzyme immunoassay helps in localization of antigen in tissues, and enzyme labeled antigen and antibody can be done. Either you can label the antigen or antibody with the enzymes. So depending upon whether it is a multiple step that you are going to carry out, or a unitary step or a single step, you can classify the enzyme immunoassay into two types, and also depending upon which type of antigen you are going to detect, you can classify them into two types: homogeneous enzyme immunoassay, heterogeneous enzyme immunoassay. Homogeneous enzyme immunoassay is a one-step process where you have all the reagents mixed together to see the positive reaction or a negative reaction. Mostly used to identify or assay the haptens such as drugs. It is not used in the identification of the pathogenic group of microorganism. And the example that is associated with the homogeneous enzyme immunoassay is the enzyme multiplied homogeneous assay technique. Now coming to the heterogeneous immunoenzyme assay, it is a multi-step process where each of the reagents or the reactions are usually carried out in different steps. You cannot carry out in a single step, but you have to add the reagent at a different steps, and hence it is a multi-step process. The best example associated with the heterogeneous enzyme immunoassay is the ELISA, enzyme linked immunosorbent assay. So coming to the enzyme linked immunosorbent assay, very sensitive test helps in easy detection of either the antigen or the antibody associated with the disease. Mostly this type of technique is used to identify the HIV antibodies that are usually seen in an HIV patient. So it is a type of immunosorbent assay. It is called immunosorbent because it helps to absorb the material, either the antigen or the antibody, towards a substrate, and these are adsorbed onto a particulate matter such as cellulose or the agarose, or on a solid phase such as polystyrene, you have polyvinyl, polycarbonate, tubes, microvells, etc. Or they can also be adsorbed onto the membranes or the disc. And here in case of ELISA, there are 96 well microtiter plates helps to identify not one or two but different types of antigen antibody interaction. So the basic technique that is used over here is to the microtiter plate we add the adsorbing antibodies, and after that we add the antigen and keep it for the incubation time so that there is an interaction between an antigen and antibody. Later we add a conjugated antibody that is linked with the enzyme, and after there is an interaction between these, we add the substrate. The enzyme is now ready to break down the substrate into a colored product. So the colored product indicates the intensity of the reaction between an antigen and antibody. Also gives us a positive or a negative test. So the variations of this enzyme immunosorbent assay is you can carry out a direct immunoassay where only antigen antibody along with a conjugated enzyme labeled antibody is used with the with the substrate, and you can go for the sandwiched ELISA technique where the enzyme will be sandwiched between two types of antibodies. First, the antibody that are fixed onto the plate will interact with the antigen. After this, the secondary antibody that is labeled with the enzyme will be added later on with the substrate that the enzyme can convert the substrate into a product. This is called as the indirect ELISA. In the competitive ELISA, first we take the reaction mixture outside this kit. We see to it that there is a binding of labeled antigen or antibody, and then we pour it into the ELISA plate and see for the development of the color. So that is called as a competitive ELISA. So there are different variations of the ELISA technique to detect either the antigen or the antibody. So it is the positive result is usually expressed in the form of a colored product. And these were the multiple step that is usually used to identify the antigen or the antibody. Nowadays we have got kits that helps us to identify these antigen or antibody in ELISA technique within a short period of time. They are called as the enzyme linked cassette. So this enzyme linked cassette can not only identify one or two, but it has got number of slides with it to identify to carry out this techniques multiple times, and helps in identifying HIV-1A antibody or HIV-1B antibodies. Next is the chemiluminescence immunoassay. So another modified form of immunoassay is conjugating the antigen or antibody to the chemical substances. And here the chemical substances that are used are luminol or the acridinium esters, and the positive result can be estimated by a signal, that is a light, that can be amplified and can be measured. So next is the immunoelectroblot technique. Blotting technique is of three types, where we blot we separate the DNA or the RNA or the protein, make it into single standard form, and we blot it on the nitrocellulose membrane. So here the same technique is usually carried out, but here we have three procedures, three steps that takes place in immunoelectroblot technique. So first we separate the ligand, that is the antigen, and then we fix it onto the nitrocellulose filter paper disc, and after this we go for the immunodiffusion. Either go for the enzyme immunoassay, or you can go for the radioimmunoassay. So this blotting technique has multiple advantages, because while going for the immunoelectrophoresis, we are going to separate the compound into different components, and then when we are binding it to nitrocellulose membrane sheet and going for the immunoassay, it is easy for us to identify even the smallest component that is involved in a particular reaction. That is among, like if you are going for to identify a particular protein, so among that particular protein also the minute component that is involved in the interaction can be known with the help of immunoelectroblot technique. Next is the immunochromatographic test. Chromatography is to separate the components based upon their density, based upon their size, particles, etc. So this immunoelectrochromatographic technique also has the advantage where the antigen is first chromatographed to separate into its component, and then we go for identifying the interaction between an antigen and antibody. So this is usually done in the form of a cassette, and the cassette has a membrane to which the antibody is usually fixed. The best example where the immunochromatographic technique is used is in the detection of HBs antigen. So here the membrane has a antibody, that is anti-HB antigen colloidal gold conjugate, and the test serum is usually added to the first window. So the three windows, that means the component usually gets separated into three windows. So it moves till it moves to the third window. In the first window we add the test serum and it's allowed to flow depending upon the capillary action. So when they move on the first window, they usually form a colored band, and which moves on till it moves the second window. So the band that is seen in the second window is taken as a positive reaction, and when there is no band that is formed, it's taken as a negative reaction. And similar type of band has to be seen in the third window, that gives a confirmation of the positive result. If there is no band that is formed in the third window, that indicates a negative reaction, because it is there where the control is usually present. So the antigen or the antibody that has to be tested is chromatograph or moves till it forms a band if there is a specificity. So that is called as immunochromatographic techniques. Next serological reaction is the use of electron microscopy and the conjugation with the immunoassays. So here the assay is usually done to identify the virus particle or a specific sera under the electron microscopy. Mostly used to identify the hepatitis A and the diarrhea virus, and the logic behind using this is the antigen and the antibody are mixed and they form a conjugate that can can be seen under the electron microscopy. And apart from that, you can go for the immunoferritin test, where the ferritin, which is a hot spleen, which is highly electrons dense material that is used in the electron microscopy, is conjugated to the antibody mixed with the test serum or the test antigen and seen under the electron microscopy. And next is the immunoenzyme test, where the enzyme peroxidase that can be used under the electron microscopy is conjugated to the antibody and mixed with the antigen. If there is a interaction, they can be seen as a conjugate under the electron microscopy. So these are the different type of tests that are involved in serological reaction, highly sensitive, easy to identify either the antigen or the antibody, which is not possible with the classical experiment like microscopy or the growth of the organism in the media, etc. So this is a unitary method or a single step where you can identify either antigen or antibody within a short period of time, and helps in diagnosis of this antigen or antibody and treatment of that particular disease after identifying whether it is the antigen that is related to particular disease or not.